Good afternoon, all. Welcome to the first webinar of the decade, 2020, end of the year for the H3ABioNet webinar series. My name is Pablo Abelchauke. I'm a training and outreach coordinator for H3ABioNet, and I'm part of a team of webinar organizers. That includes myself, Faisal, Sumi, Aza, Nikki, and Wisdom. I hope I don't forget anyone, but we are a team of webinar organizers, and Amel as well, by the way. So we are a team that makes sure that on a monthly basis we have a webinar taking place. And the theme for January's webinar is Genomics, as you'll hear throughout the presentation. And we are honored and privileged to have invited two speakers from the Broad Institute. The two speakers, I'll just read a bit of their biographies so that we have an idea of who they are. But you can Google them. They're findable on Google, and you can find out more about what they do and who they are. But just for now, so that you know who's going to be presenting for us, the two speakers are Dr. Eric Banks, who's a computational biologist, institute scientist, and senior director of the data sciences platform at the Broad Institute. He leads an amazing team of computational biologists and software engineers that implement novel computational methods, engineers and operates large-scale production pipelines, and develops innovative infrastructure to handle the Broad Institute's growing computational needs. The second speaker is Dr. Geraldine van der Owera, who is the Director of Outreach and Communications for Data Sciences Platform, DSP for short, at the Broad Institute of MIT and Harvard. As part of her outreach role, she serves as an educator and advocate for researchers who use DSP software and services, including GATK, the Broad Institute industry-leading toolkit for variant discovery analysis. And the title for today's talk, as you can see on the first slide, is Genomic Analysis with GATK, Best Practices and Latest Innovations. So just a bit on the abstract. Genome Analysis Toolkit, or GATK for short, was developed at the Broad Institute as the most widely used open source software package for variant discovery on whole genome and exome data. In this webinar, Eric and Geraldine will provide an overview of the current state of the art in variant discovery analysis for human medical population, genomics. They'll start with a short introduction of the toolkit and its requirements. Then they'll swiftly proceed to outline the GATK best practices for the most common use cases, which will consist of calling short variants, SNPs, and Indels, as well as a copy number variants in both germline and somatic contexts. So if you want to read more about that, we sent an ad out, so you can read more on the abstract, but they're going to continue with the talk soon. Just a bit of housekeeping. This talk will be about 40 to 50 minutes, followed by 10 to 15 minutes question and answer and discussion session. So if you have questions, can you please type them? I'm going to point my mouse, if you can see at the bottom here where it says hi James and Fozia, you can tap your question there just to make sure that we don't disturb our speakers. Again, if you want to speak at the end, you can raise your hand. There's an icon on top next to the camera icon. There's an icon with a human being raising their hand. You can click on that, and myself or the speakers will recognize you. And yeah, without further ado, I would like to say thank you so much to Eric and Geraldine, who we've had it back and forth since November last year, preparing for this webinar. So this is truly highly anticipated, and we're looking forward to learning a bit about GATK, best practices and latest innovations. Eric and Geraldine, the mic is yours. Welcome to the floor.
Thank you so much. Thank you everyone for joining us this morning. Well, it's the morning for us, probably later in the day for you guys. Okay, so we're really happy to be with you to talk about GATK. We're going to structure this talk in two main parts. In the first part, I'm going to give you an overview of the toolkit and the best practices at a high level. And then in the second part, Eric will talk about some of the more exciting work that's being done by the development team. So I will start with the overview. If I can change the slides. Yes. Okay, here we go. All right. So GATK stands for Genome Analysis Toolkit, and we're currently on the 4th version. So we released this version in 2018, and I can't believe it's already been 2 years. GATK4 is a big rewrite of the original tools. It's been re-engineered to be very scalable, very robust, and as fast as possible. Now, the scope of work that you can do with GATK is mainly focused on variant calling, but as you'll see, we've been extending that beyond its original scope. And one thing that's very important, because this is a toolkit with a lot of tools, I think at this point there's a couple hundred tools in the toolkit. We also provide best practices workflows, which are guidelines for how to string multiple tools together to get the best results possible for a particular use case. And so today I'll cover the 3 main best practices workflows that we have. The toolkit is completely open source under a BSD3 license, so it's completely free for you to use and adapt for your needs. And so let's talk a little bit about what this toolkit allows us to do. Right, so we're going to be looking at genomic variation, and we know that genomic variation can take many forms, from the very simple short variants that we have, which can be simple nucleotide changes or a few nucleotides that are inserted or deleted compared to a reference, to more complex events like copy number, where you might have a region that is deleted or that is duplicated, and so there's different numbers of copies of that particular region. And that's going to show up as differences in the amount of coverage in the sequencing, as we'll see in a moment. And then there's even more complex kinds of events like structural variation, where you can have major rearrangements, you can have inversions, translocations, and so on. And so these are the main types of variants that we're going to be looking for. In addition to that, each of those comes in 2 flavors. They can be germline or somatic. In the germline cases, that's the variation you inherit from your parents. And the somatic variation is variation that arises during your lifetime. We often think of somatic variation principally in the context of cancer, but there are other contexts where somatic variation arises as well. However, in this talk, we'll mainly focus on the cancer genomics use case. So how do we identify this variation in people's genomes? We're going to sequence the genome, and by sequencing, we're going to generate huge piles of data for each individual. And that, of course, leads to certain complications. So when you're sequencing a genome, you're generating this huge haystack of sequencing data. And within that, you're not just looking for one needle, you're looking for millions of them. Because you'll find 4 to 5 million small variants just from looking at the germline short variants. And so ultimately you'll have the difficult task of deciding, of those millions of small differences, which ones actually matter, whether you're looking to diagnose a genetic disease, whether you're looking for associations with traits, or just looking at the evolution of populations. You're going to have that very complicated task of identifying which variants actually matter. And of course, it's crucial for that work to have a really high quality call set of variants to start with. And that's what we're going to focus on. So how do we do that? That's where the GATK Best Practices workflows come in. So for each major variant class that we've just reviewed, there is a GATK Best Practice workflow that provides guidance on what are the data transformations and the data analysis steps that you need to perform to identify this type of variants with high level of accuracy and precision. Now currently we have production level workflows, meaning fully validated workflows for short variants, which are the SNPs and Indels, as well as copy number in both the germline and the somatic context. We also have a germline structural variation workflow that's currently in development, but that's not fully ready for production yet. If there are questions, interest about the structural variation, Eric will be happy to discuss that during the question time. I'm actually going to focus on the short variants in germline and somatic cases, and copy number in the somatic case as well, with a brief word on germline copy number. Now, these workflows, I should say that the best practices are primarily guidelines, and there are many ways that you could validly implement those guidelines into your own workflows. However, we provide reference implementations that are actual scripts that you can use out of the box if you're interested. They're written in a language called WDL. WDL stands for Workflow Description Language. And we publish these workflows in GitHub at the address that you see on this slide here, up here. We also provide a version of these workflows that are fully loaded with example data and that work out of the box in a cloud-based platform that we operate that's called Terra.bio. And this platform allows us to provide workspaces where each workflow is pre-configured with some example data, so you can go in and you can try it out, you can test it out without having to do any installation on your local system. We really encourage you to check this out, even if you don't plan to use this for your day-to-day work, because you have infrastructure available, because cloud is not an option for you at this time. That's not a problem. This is really intended to allow you to try out the pipelines, to see it's working before you invest the time and effort into installing the pipelines on your own systems. Okay. Now, I should also mention we've recently started a partnership, a very exciting partnership with a team at Illumina. They have a hardware-accelerated genomic solution that's called Dragon. And our teams are now working together to align on the same, co-develop the same methods and ultimately arrive at the same best practice pipeline that will be called Dragon GATK Best Practices. And the goal is to be able to provide a single pipeline that will exist in both the regular software implementation that's fully open source, completely free, that we maintain and distribute, that will be completely functionally equivalent with the hardware accelerated implementation that is provided by Illumina. So that whether you choose the accelerated implementation because speed is the most important thing to you, or whether you choose the free open source version, the results will be functionally equivalent, and you'll even be able to cross-analyze data sets analyzed by the different systems. And so the goal is really to give you more choice and flexibility when you choose a pipelining solution. And we're, of course, more than happy to address questions about this during the question time. All right, with that, let's dive into the actual GATK Best Practices workflows. This is going to be an overview at a very high level. There's a lot of ground to cover. We have extensive documentation, educational materials, workshop materials if you'd like to take a deeper dive into the specifics of what are the tools and how they work. Here what I want to do is really give you an orientation to what are the key concepts in each case. For each use case, what are really the important concepts that you need to understand in order to get started. Regardless of the type of variants that you're going to analyze, you're going to need to apply some data pre-processing to go from the raw reads that are produced by the sequencing machine to get to the point where the reads are ready for analysis, for the variant discovery analysis, there's a number of steps that you need to apply in order to prepare the data. And the main steps that are listed here are mapping, of course. Initially, you just have read sequence, and you need to first find out where that read sequence maps to the genome, where it belongs in the genome. And then the marking duplicates and base recalibration steps are two pre-processing steps that are intended to compensate for some technical artifacts that are inherent to the sequencing process. And so these are going to be cleanup operations, so that when you come out of this data pre-processing phase, your reads will be nicely mapped, cleaned up, and ready to be put through the variant analysis as such. So with that, we can dive into the germline short variance workflow. For germline short variants, which are going to be your SNPs and Indels, the really central concept is this idea that joint analysis empowers discovery. And what does that mean? It means that you're almost never looking at a single genome by itself when you're looking at germline variants. Regardless of what you're trying to discover, you typically need to use family context or population data to build your analysis. If you're trying to diagnose a child with a probable genetic disease, you'll want to have sequence information from the family. If you're looking at traits in different populations, that is definitely something where you'll need a large number of individuals in order to do that kind of analysis. And so in every case, you'll have multiple people's sequence. And we recommend not looking at this as individual data sets where you just call variants for each person and then put all the variants together. For a number of technical reasons, that leads to an underpowered analysis. So instead, we recommend doing what we call the joint call set, where you're going to do an analysis on all of the data together as soon as you can. And that's going to give you a lot more confidence and power in your analysis. Now, in practice, how does that work? We have a workflow that we call the GVCF workflow that basically operates in two steps. The first step is you're going to do a variant calling per sample, but in a very particular way. We use a GATK tool called haplotype caller with a special mode we call GVCF mode. A GVCF stands for Genomic VCF.
Apologies. I think we might have lost them. So if we can wait for 2 minutes while we try and reconnect.
The Wi-Fi dropped. Okay. All right. So we were talking about GVCF, genomic VCFs. So the idea here is that the genomic VCF allows us to capture information about the genome, record what sites we think might be variants, but also record important information about the sites where we do not see obvious variation. And so we're going to capture information across all sites in the genome in this particular format called the GVCF. We do that for each sample, and then once we have GVCFs for all of the samples, we're going to do an operation, first a just purely technical operation of consolidating the GVCFs into a sort of database. And then the actual interesting analysis, which is the joint calling of the cohorts, where we're going to look at the GVCFs across all of the samples, take all the information we have available into account, and produce the final call set that will be a multi-sample VCF with information for the variance of all individuals in the population that we're looking at. And so that's really the key, the key methods in germline variant calling for short variants. Now, what you produce with that is a raw call set, and it's important to understand that the variant caller is designed to be as sensitive as possible. So we know that there are some things that will be called that are false positives. They're not real variants. But we want to include them in the first pass because we want to give ourselves the flexibility of doing filtering passes and refining the call set afterward. So when you get that raw call set out of the joint calling workflow, you then need to apply some filtering tools. If you're working with a cohort, you're going to use a method called variant recalibration, which involves a couple of tools. And variant recalibration is going to allow you to use machine learning techniques to filter and classify your variants, give them a score that represents their likely likelihood of being true variants as opposed to false positives. And then that gives you a very flexible way to filter them based on the level of confidence that you want to have for the variants that you continue with in your analysis. And this is a procedure that is that largely automates the modeling and the filtering of the variants. If you have only a single sample, a very small call set, that doesn't typically contain enough information to do variant recalibration, and so you're going to use a different set of tools. It's also using machine learning under the hood, and in this case, it's a special kind that's called deep learning. It uses neural networks to model the variation and then produce essentially classification of your variants as well. Now, I know it sounds complicated. Neural networks is something that has been getting really popular recently. The implementation that we have in GATK is ultimately quite easy to use because we've wrapped the modeling in the GATK tool so that ultimately what we're going to use is just GATK command lines that look like any other GATK tool command lines. And that's why I include some on the slide to show you that these are completely usable by anyone. You don't have to be an expert in machine learning to use these effectively in your research. Okay, so that's for the single sample case. And that really concludes the key points that you need to know about germline short variant discovery with GATK. So now let's move on to somatic short variants. Okay, this looks like a complicated workflow, but ultimately it boils down to a couple of core concepts. And specifically in the concept in the context of cancer, the key thing that we're struggling with here is that when you look at the tissues, the cells that are inside a tumor that's evolving, there's a complex genetic landscape because as the tumor evolves, new variation events occur, new mutations occur. And so if you imagine that you have an initial event that kind of sparks the development of the tumor, and as it progresses and more events accumulate, you have different subclones that start evolving and proliferating within the tumor. And so in the tissue context, that means that you will have a heterogeneity of the genetic makeup, the genomic makeup of the different subclones. And so when a pathologist is taking a biopsy of a tumor, for example, ahead of sequencing, it is very rare that they'll succeed in getting pure tissue sample. So what you'll get is a mix of different cell lines, effectively. And so when we sequence that, it's the amount of evidence that you'll find for each mutation, for each variant, is going to be mixed in and diluted because of this heterogeneity. So that really complicates the picture, and that's why you need a different set of tools compared to the germline workflow. So the fundamental concept that we're going to apply for this kind of analysis is the idea of the tumor normal comparison, where if you have a normal sample from a healthy tissue from the same individual, you can use that as a baseline so that when you're looking at the tumor, first you can subtract that germline background, okay? So you have the germline that shows you where you probably have germline variance. And then when you look in the tumor, you can say, well, this is probably not somatic. This could be somatic because I see some differences in the read data that is not present in the normal sample. So that's your starting point, is this tumor normal comparison. However, that's not sufficient because you also have a number of artifacts that are going to affect the analysis. You can have recurrent artifacts and data quality issues that will make it look like you have somatic variation in your tumor because it's not present in the normal, but it's actually sometimes due to the way the tumor tissue is extracted and preserved can introduce some of these. In addition, you can sometimes have germline events that are present in the normal but not discovered when it's sequenced. And so you could think that this is a somatic mutation, but it's actually not, it's just germline. And so to deal with both types of artifacts, we're going to use two key resources. One is the panel of normals, which is a resource that you compile by taking normal samples and putting them through a somatic mutation workflow to collect essentially all of the low frequency things that look like variants but are actually artifacts. You collect that across many normals, and that allows you to filter out just these recurring artifacts. And you also use population resource that gives you kind of the baseline germline to try to eliminate any germline events that might not have been correctly identified in the matched normal, but that is present in the population, and you can use that as a basis to eliminate those sources of false calls as well. So those two resources are going to be very important. Now, if you don't have a panel of, if you don't have a matched normal, it is possible to run the workflow, but that does make the panel of normals and the population resource even more important as a result. Okay, so that's those are the key concepts for somatic short variants, really understanding the complex nature of the heterogeneous environment of the tumor and this use of resources to deal with that. And so we can move on to the somatic copy number alterations, CNAs, where we're looking for evidence that there are additional copies or fewer copies of a particular stretch of DNA. And the idea that's going to help us identify that is that if you have a duplicated region somewhere, and let's say this gene TP53 is duplicated and is present in multiple copies in the genome, then when I sequence this genome, I'm going to find more reads coverage for that particular region. If we can assume that when we do the sequencing, we have roughly the same amount of sequence for every piece of DNA that we put into the sequencer, if there's a duplication, we should get twice as much coverage out of the sequencing process. So that's the core idea. Now, the complexity of that is that we know that there are biases that can produce more coverage in certain areas of the genome, and so we'll have to deal with that. But that's the underlying idea. And so with this idea, we're going to say, okay, we're going to collect the coverage. We're going to count how many reads overlap each target. If you're looking at an exome, you'll look at exome targets. If you're looking at whole genome, you can chunk up the genome in bins of a particular length. And for each bin, you just count how many reads overlap that bin after the mapping process. Then you'll have a step to normalize the data to compensate for those variation in coverage. And I'll show you one more slide about that in a minute. And once you've done that, you can identify segment boundaries. And so this is what it would look like if you have each band, each vertical band represents a chromosome, and each dot on this figure represents one target or bin. Okay? And so here we're plotting the copy ratio, which is how much coverage do I have at this location compared to the rest of the genome. And once you've done the normalization, you should be able to see that when there's no copy number event variation, your dot should align around 1. But then if you do have, for example, a duplication, in this case we have a duplication of the chromosome, of the entire chromosome, then you have this big jump, and you see it reflected in the total copy ratio. And so that way you can identify these events. Now, as I mentioned, there are biases that lead to different amount of coverage being collected in different areas. And the good news is that there's a systematic aspect to this. And so we can actually normalize, and I won't go into the detail of the method, but we have a lot of documentation about how that works. But basically, through a process of normalization and refinement of the data, we can go from a picture where the dots that represent the raw copy ratio are really smeared in a way that you couldn't possibly identify any real events here. It's impossible to guess what's happening. But as you go through the normalization process, you can see that the signal really tightens to the point that you can identify segments.
Sorry, if you're not speaking, can everybody please mute their mics? There's a feedback, sorry.
Yes, thank you. Okay, thank you. All right, so so the point is here we are narrowing down, cleaning up the signal to the point that we can recognize even smaller events and make calls about whether some regions are amplified or deleted. So that's the key process and key concept involved in somatic copy number variation discovery. Okay. Finally, just a short word on germline copy number variance. This workflow is production ready. We just haven't had as much opportunity to document it thoroughly. But we have recently hired somebody to help us with the documentation and who is going to make a big difference here, so no pressure, Derek. But basically, germline copy number is a workflow that's similar in concept to the somatic copy number in the sense that you're looking to identify these jumps in coverage, in copy ratio. However, in the germline context, the data looks a little bit different, so we're also going to take advantage of having many samples, looking across many samples to do this. And the implementation is a bit different in the sense that it's a machine learning model that does the entire process in a single step. So the denoising, segmentation, and calling are all done simultaneously. It's actually a little bit simpler to operate. But we'll have more details coming out about that soon. All right, so that covers our main GATK Best Practices workflows. As I mentioned earlier, structural variation is a work in progress, and Eric can answer questions about that during the question time. One more thing before I hand the mic over to Eric to talk about the latest innovations and what's coming down the pipe is a shameless plug. I've been working on a book for O'Reilly with a collaborator, Brian O'Connor, to describe all of these workflows, how to operate them, specifically in the context of the cloud, but there's a lot of detail about how the workflows work, the logic, how they're implemented, and that will be coming out in May 2020, if you want to check that out. And with that, I will hand the mic over to Eric, who is going to talk about what to expect in the near future from GATK team.
All right. Thank you very much, Geraldine, and thank you to everyone out there. It's an honor to be here presenting to you. All right, so as Geraldine mentioned, I'll take care of a few new initiatives here. This is a little bit different than the genomic stuff that Geraldine presented just now. And as you see, the GATK team is trying to branch out a bit and trying to help more in wider various contexts for the genetics world. All right, so the first one that I want to kind of talk about is this single cell sequencing. For these, I just don't, because it's not classic GATK, I do take one or two slides for each section here just to explain what this stuff is. For those who don't know, generally genomics, when we sequence a genome or an exome, we take the cells, throw them into a big blender, mix them all up, and you get all your cells together in one big mush, and that's that nice Kiogen, image that we like to steal and show everyone, that's the fruit smoothie. They're all in a blender. Whereas now you're able to select out single cells. So specific, you can go to a specific tissue and pull out a cell from that tissue and know that it comes from that tissue, and then sequence that particular cell. In this context, the most classic way of sequencing is RNA sequencing. What you're looking for there is just expression levels of different genes. But now you can tell the expression levels of a specific gene in a specific tissue type, which is actually very powerful. And what I'm showing you here is just a random t-SNE plot. I only show you that because it is, we'll use it later on inside this particular presentation. You don't have to see the details here. All you have to know is a t-SNE plot is kind of what people use in the world of single cell when they have divided up their cells, and they figure out based on the expression patterns which one, how the cells divide up among different tissue types. So really, if you've ever done principal component analysis, PCAs, and you've separated into different principal components, you can imagine it looking the same way here, where you've split up your different cells by tissue type. And that should easily come out based on the expression profiles of your individual cell types. Now, we've got a new software package. I'll note that I call it a GATK extension here, and that's because it's not in the GATK proper, mostly because it makes heavy use of machine learning modules, and it was better and more easily implemented in Python. So it's certainly under the GATK umbrella, but it's called CellBender. For those of you who want a bit of a history lesson, when we were developing GATK4 2 to 3 years ago, the code name of that project was Hellbender, the lead developer there loved salamanders, and a hellbender is apparently a very large salamander. So with a play on words here, the single cell team has nicknamed themselves the Cellbender team, and they've gone live with that as the software package name. But it is under the GATK umbrella nonetheless, and because of that, it means we have good documentation. It's all open source. As Geraldine mentioned before, all these workflows are available in Terra, so people can just take them and use them as is, or test them with test data, and they're even working on papers to describe this. Now, I want to correct one thing that Geraldine mentioned earlier. It turns out back in 2009 when we started the GATK, the variant caller wasn't the first tool we created. It was actually the data processing tools. I think the theory was, we didn't realize that at the time we were doing this on purpose, but you must really understand the data before you can really start variant calling and creating a tool to do variant calling. And that's why most of the tools we created initially were there to really understand the data, and when we found issues with it, to go ahead and create tools that reduce the noise. That's a lot of what Geraldine showed in the last 20 minutes. And so that's really what Cellbender is made up of now, is a bunch of tools to denoise the process. I'm just going to show you one of them, and I chose this one because it's got the coolest name. And we've been people have claimed we name things terribly in the GATK. They're not splashy, and that's probably my fault. But this one has a splasher name called the Droplet Time Machine, and it's also easier to show you what it does. So again, generally what you're doing in single cell is you just take a look at your genes and the number of, and you want to just look at the number of reads there, and that will tell you the expression profile. And it's simply because you're doing RNA. So if a gene is expressed, there will be RNA there, and when you sequence it, there will be reads. And if a gene is not expressed, there will be no RNA, and there will be no reads for that. The problem with that is that's a very naive approach, and so the Droplet Time Machine tool in Cellbender makes use of reads to remove noise, and that's not what the classic tools do. And I'll explain that much more clearly in this slide. So what I've done here is I've just taken a run of single cell sequencing, and I've made that standard t-SNE plot I showed you before, but the coloring here is different. Now I'm coloring it based on the amount of expression of a particular gene. So if you look at the LYZ gene, that little cell, for example, you'll see that the cells have been broken out into their different components. And what you'd expect is that particular cell types will have high expression of LYZ, as you would expect, and all the other ones will have no expression. Either the gene's on or it's not on. And I hope you can, this is rendered in high def enough for you, but you'll see that there's a cloud on the right there with high expression, nice dark purple, and the rest of it is not gray, it's purple-ish, which is not good. It means that there's low expression of the LYZ gene across all the cells and all the tissue types. And that's not what you'd expect, and that just can't be right. And that's the noise. What you're seeing there is the noise in the process. The fact that it's not gray, it should be either very gray or very purple, and it's not there. And what the Droplet Time Machine tool does is model that with some machine learning modules, and it's actually pretty simple. But what you see afterwards is this. And those are those same plots now after gone through the denoising process. And you'll notice that there's a very dark purple cloud, and now most of the other clouds are pretty gray. And that means that the expression levels are more accurately reflected here, and the noise has been cleaned out through the process. So that's just one example. I just wanted to highlight, my job here is to highlight, get you excited about some of the new stuff coming. This stuff is all available on GitHub. Documentation is there, as I mentioned. So anyone doing single-cell sequencing should please take a look at it. We use these tools ourselves in our production runs. So these are ready for prime time, and every month or every quarter or so, we release new tools in this context. And it's a very fun team. All right, now I'm going to jump to a whole different topic. We in the GATK, in the early days, we had support for reads other than just standard short Illumina reads. Those as the sequencing technologies themselves became less and less common, the support for them dropped out of the GATK. But now we've started to add support for longer reads. So in this particular image, you see PacBio, Pacific Biosciences, Oxford Nanopore. Those are long-read technologies, and we hadn't really supported them well in the GATK, but that is changing. We have a whole team now. The slide I'm showing you now is just internally. I grabbed it from our lab. The most important thing I want to point out is on the bottom panel, you see the length of the reads. So for PacBio reads, they have an average length of about 100,000 bases. So again, with Illumina, you're talking about between 100 to 300 bases. Now you're talking about 100,000 base long. That this is what we mean by long reads. It's not 1,000 bases; it's tens of thousands of bases. And really, for PacBio, they have these corrections, which I'll show you in a bit, that bring the size down to 10,000 bases. But 10,000 bases is still pretty impressive. And similarly on the Oxford Nanopore machine. Okay, so one thing that PacBio does is it takes a molecule, and instead of just making a 100,000 base long read with very high error rates, the problem with the long read sequencing now is they're not as accurate as the short reads, and there's a lot of errors in the bases there. What they do is they just sequence in a circle over and over again. So they sequence the same 10,000 bases 10 times rather than 100,000 bases one time. And then they take a consensus across those 10 different circular runs and improve the accuracy of the reads. And that generally works really well when it works. You get high accuracy. When that happens, you get reads that are of equivalent accuracy to Illumina reads, but obviously much, much longer. The problem is that very often it doesn't work. Either you haven't gone enough around in the circular consensus, you see in this picture, or sometimes you have these poly A tails that just fall off, and for some reason the software with PacBio just doesn't allow you to run a consensus for that. And it turns out you lose a significant chunk of your data that way. Unlike Illumina reads, where sequencing where you have hundreds of millions of reads for any given run, here you have thousands to tens of thousands of reads, and if you lose half of those, you're losing a lot of your data. And so one of the big tools we've built goes ahead and tries to correct that and recover all your data. So this isn't, so PacBio is the one that has the software to remove the noise, but we have the software that comes along and recovers a lot of that noisy data that PacBio just generally throws away. And the concept is actually pretty simple. If you take the reads that succeeded the consensus calling and use those as a scaffold to help build the consensus from the other reads that they've thrown away, it's actually pretty easy to go back and recover those reads. And that's a tool we've built. What I'm showing you here is very simply a kind of a schematic of before and after. On the top, you see all that purple. Now, I want to remind you that these are tens of thousands of bases long reads. So when we kind of trying to show you a read here, this is an IGV type screenshot, all that purple are mistakes in the reads. And you'll see lots and lots of mistakes. There's so much purple and so much color because there's so many mistakes in these tens of thousands of bases long reads. But when we've applied the cleaning and the consensus and the recovery, you'll see now we've pared it down to just a few reads that are very high fidelity, very high accuracy. Those little colors on the bottom panel that are in there are meant to be there, the real variation across many thousands of bases. And so it's a pretty cool tool. So for those of you running long read sequencing, we have it all set up.
Thank you. Yeah, I think there might be an issue with Wi-Fi. We'll try to fix that. So again, apologies. There might be a bit of connection issue. Eric?
Oh, all backwards is terrible.
Falling apart.
Falling apart. All right, so I'm actually almost done. I want to leave time for questions. So I will just run through this and say there's a very cool project at the Broad, not ours. The PIs are listed below. That run rare genomes for people generally kids with rare diseases, where they sequence the parents and the kid to try to figure out what's going on. We have developed a long read pipeline for that, where you do long read sequencing on the child, short reads on the parents, and it's actually, I just point out, obviously, just like with the normal GATK best practices for genomics, it's not all our tools. We're just trying to put together the right pipeline that maximizes the results and the science for everyone downstream. In this particular project, there was one case where they really wanted help. We piloted this whole long-reach project, and it was pretty nice. They found the causal variant for the child. This child where they didn't know what was going on. They had a phenotype, but they couldn't figure out the genotype. And through long-read sequencing, they found a large de novo event, a very large deletion that wasn't clear from short-read sequencing because it was in a very repetitive genomic context, and that just doesn't work well with short-read sequencing. I wanna leave time for questions, so I'm just gonna jump very quickly for the next few slides and say that people have complained in the past that we focus too much on humans. Geraldine herself has complained to me. That's her background in non-human genomics. So just to say, we've gotten a couple grants to start in 2020. So for the next 2 slides, we have not done any work yet, but the plan for 2020 is to start working to extend and expand the GATK to handle non-human organisms more elegantly. Right now, people kind of push the square peg in the round hole. But one is on the malaria side, where we're going to work better to process both parasites and mosquitoes. And the other one is we got a grant from Chan Zuckerberg to do GATK for bacteria, which hopefully isn't a lot of novel tools, but more an improvement of the current tools to expand and include the flexibility to handle non-human organisms. And with that, I want to end just to leave time for questions. I'm sorry again that the Wi-Fi cut out a couple times. Thank you for listening, and we're very happy to take any questions.
Okay, sure. Thank you so much, Eric and Geraldine, for that presentation. We have about 6 minutes for questions. So if you have any burning questions, you can raise your hand using the icon on top next to the camera, or you can type in the chat box, and Eric and Geraldine will definitely answer your questions. So if you have any questions, someone has typed. I'm going to move the box so that we can read. Wisdom has a question. Can you use GATK to call variants from BAN files aligned with a different software? Secondly, can you use GATK to produce VCF files from VCF files called individually? If not, what will you recommend? That's a question from Wisdom. Eric or Geraldine, may you please answer that question?
Yeah, I think we both know the answer, so I jumped in and took it first. So the first question is, so can you call variants on BAM files along with a different software? So the aligner actually isn't even GATK software. The aligner we use in our production pipelines is BWA, but there are many other aligners and GATK. So once you've aligned and created a BAM file, GATK works on any and all of them. It doesn't really matter which aligner you use. Again, we use BWA, but it's not even our software. It's developed by Hung Lee, who's now at the Dana-Farber Cancer Institute. As far as if you already have it, so to create a GVCF file, you need a BAM, you need the read data. A VCF file only lists the mutations, and you can't go from a VCF to a GVCF. You need to go back to the BAM file and create the GVCF there. The reason why is the GVCF, again, contains not just the variants, but also the non-variants. It's very important that it records the places in the genome where the sample is confidently not variant and has high reference confidence, because that's important for that joint analysis that Geraldine mentioned. And to do that, you need to see the reads and see that there's no variation in those reads. So what I would recommend is go back to the BAM and create a GVCF from that.
Okay, thank you, Eric, for that. Wisdom says thank you. And there's a second question from David. For the CNV discovery germline, instead of including multiple individuals, is it possible to explore machine learning methods that compare target sites with known CNV neutral regions in a single genome?
So certainly, I mean, that's basically what that panel, so when Geraldine talks about the panel of normals, both on the somatic and the germline context, that's basically what that's doing. It's taking as much knowledge as you can from the rest of other samples and applying it to your sample here. So of course, if you only have one sample, you could do your best based, I mean, what you're saying, the suggestion here is known neutral or no non-neutral sites. That's exactly what the pen of normals is. So, I mean, it's a good suggestion, and I think that's exactly what we're trying to do. There's another question here. Yeah.
There's another question here from Hussein, who says, are there any plans to develop tools for genome graph-based analysis?
Yeah, that's a good question. So absolutely, so that long reads analysis team, that's one thing they're aiming to do. I personally, now this is very subjective. I'll say this. I still think we're far away from people being able to easily use graph-based assemblies. The other tools downstream just aren't ready for that. If you think about people doing GWAS and other downstream uses of it, they're just not set up to handle graph-based references, and that's important. But that doesn't mean we're not going to try. And the long reads team is very, very much engaged in those conversations. At least the NIH in the US has sponsored a long read sequencing new reference consortium to try to build graph-based references, and we're helping out with that as much as we can. So there are plans for that. I just don't want to promise that it will work in 2020.
That's fair.
Okay, David says thank you. I see people are typing, so I'm gonna wait for them to. Okay, so Hussein also says thank you as well. But just so the participants or other attendees know, we're recording this webinar, so we're gonna make it available on our YouTube channel. So if you missed any parts or explanations in the presentation, you can easily access it after a week or so, because we're gonna upload it onto our YouTube channel. Wisdom asks another question: is it recommended to merge VCF files and create a bed file and use that bed file to then call variance from the BEM files? That is, can you use GATK to call variance based on a bed file?
So if the question is, can you use a bed file as an intervals list to basically say, I have these variants of interest, and here's a bed file that describes that list of positions, you can certainly use that as a list of intervals of interest if you want to restrict your analysis to just that part of the genome. Now, you have to keep in mind that whenever you're restricting your analysis to a subset of the genome, you might be limiting your options for later on, because if you're looking at a very small territory, then that will probably not be enough data for the machine learning filtering tools. But if you're looking at a very specific subset of positions and you're planning to look at them like visually and explore the data for every one of them, that should be fine.
Yeah, I have something to add though, which is maybe not what you're asking here, but the somatic copy number caller actually takes in the germline SNP calls as an input. And it uses that to look at different allele ratios. And so if you're asking here, can you improve your CNV calling based on the allelic ratios of the SNPs, that's part of the somatic pipeline. That might not be what you're asking, but it is. We definitely have. We do use the SNP inputs into the CNV calling because again, with copy number, you do expect a non-50-50 ratio on the germline side.
From Nast Node, can you go ahead and ask? I've approved that you can speak. Hello.
Hello.
Yes, we can hear you. Go ahead.
All right. Please, in the course of the presentation, he mentioned about how to recover some lost data. And we know in long-read sequencing, there's this common knowledge about 10% to 20% error rate. Is it the data that the pipeline is able to recover, or in the course of using the pipeline, maybe some data could be lost and they have a way of recovering it? Thank you.
It's a good question. I don't know the answer. So there's so many reasons that you might lose data along the way, from things like contamination to just bad library preparation. So there's no one, unfortunately, there's no easy answer to that question. What I showed you is the fact that even when your sequencing goes well in the long-read data with PacBio, you still might lose data because the PacBio software is imperfect. So in that particular dimension, we can go and recover pretty much all of those reads. But there's many other reasons why you might lose reads, so it doesn't mean you necessarily get back all 10 to 20% or more of the lost data. And it's certainly true. I would say on the short read side, I know definitively there's so many things that go wrong. Things like contamination and just bad library prep, it can introduce a large variability in the amount of data that's actually usable.
Sorry, thank you so much, Eric, for that. I'm reading Shakuntala's question about is it possible to have a copy of the slides? And I was saying we will ask for your approval and perhaps then email them. But there's a question from Fozia, who says, does sequencing allow better coverage and better accuracy?
Yeah, circular sequencing is actually less coverage but better accuracy because again, it's basically you've seen the same thing 10 times over again and you know it's real. So, technically it's less coverage, but you've raised your accuracy to pretty significantly high. And of course, you can have the slides.
Yeah.
These and others.
Okay, so I'll make them into PDF and I'll email them I'll look at the list of attendees of this webinar and we'll email it as well as the link to our YouTube channel, okay, Shakuntala. One more question, because we're pushing it now. We're 5 minutes after 4, so because this was planned to be an hour. So if there's any question that's burning, quickly ask it. Are you also planning on developing tools for 10,000, I think, genomics data? That's from David.
Yeah, we have that. We have tools. We have tools there. I believe there's even a whittle that Geraldine described in Terra that processes single cell data that's from 10x. I should double check that, but there's a 10x pipeline.
Well, we have a pipeline from the Human Cell Atlas project, the Optimus pipeline, that does that. I ran it yesterday.
So we definitely have it there. So yes.
But it's not a GATK pipeline.
No,
proper.
but it's available.
But it is highly validated. There's a really neat validation report and everything. We can send you the information about that. That helps.
Yeah.